afternoon. It is a pleasure to introduce today's speaker, Xabier Lopez. For many of you, he needs no introduction, but for the sake of the rest, I will mention that he started his career by doing his undergrad in Universidad Autónoma in Madrid. That's actually just now I learned that is where he got his affection to quantum chemistry, which continued later by coming here, or actually returning to the Basque Country to do his PhD in quantum mechanical electronic structure calculations. If you look at what he did during these years, it is nothing but impressive, and that's actually won him a distinction award for his PhD. After that, he did a long postdoc for 4 or 5 years with Marek in Karplus. And so if you look at his career, throughout the years, he showed quite a loyal attitude towards quantum chemistry, but I would say that it is during these years with Karplus that he was willing to I would not, but maybe to say, he was willing to show some signs of defecting into a framework in which electrons are not considered totally independent. And that was with his QMMM with Karplus, a subject that won Karplus the Nobel Prize in 2013. Later, he continued that with another postdoc with Graham Richards in Oxford. That was just after that was the time to come back here. And in 2022, he's catedrático in physical chemistry here at the university. The shift from quantum chemistry to classical mechanics was gradual but consistent, and today that is the subject of his work, his personal perspective on simulating biomolecules. So you go ahead.
Thank you very much, Froden. Well, first of all, thank you for coming here. I see many known faces in the audience, and I really appreciate that. And so what I thought to present is the IPC seminar, which I think the main objective is to give to the IPC community what is our research about. And I decided to divide this talk in the following four subjects. First, an introduction of who we are as a group, as the theoretical chemistry group made. Then I will give like a gentle introduction, I hope it's gentle, to protein simulations, just to see what are the challenges and what is the perspective of this field. Then I will introduce some of our recent work, which is more related to the MD part, to the molecular dynamics part of short peptides. So we try to use also quantum mechanics, but mostly we are interested in the dynamical problem of this system, the biochemical system, and finally some. So I hope that you have a general idea of how to locate us and what we do. Okay. So first, who we are. We call ourselves a theoretical chemistry group mainly, and it was founded by Jesus, who actually was the one that put me on, even before going to Madrid, he taught me physical chemistry at the third year, and was the one that got me interested in quantum chemistry. And the group has really grown immensely in the last years, and we combine many different fields inside the theoretical chemistry. We also even have people that are experimental physical chemists, like Lucas and Carlos, and this really have created a community that is really very interesting how to interact and how to share projects and how to learn from different fields inside the physical chemistry. If you want to know more in detail what we do in this community, we organize every 2 weeks. Daniel Reta, who is here in the audience, these KP seminars, so you are welcome to come to see what we do in the different fields of our STEM. But within this group, some people, we are working on bio stuff, on molecules of biological interest. And there is a biological research line that I co-direct with, along with David Sancho. And we treat like several systems that have in common that they have some biological interest and biological relevance. Mainly we treat proteins, okay? We also have some, actually a couple of tests were defended last year related to DNA interactions with ligands. We also treat some projects related to catalysis, but mainly today, later, I will speak on this topic here about intrinsically disordered peptides and proteins, okay? So if you see all these different projects, mainly are object of interest, not only because I said that here DNA we also treat, but mainly our object of interest are proteins or peptides. So I thought that in the spirit of this IDP seminar, I will first make a gentle introduction to protein simulations, but basically, as I said, focus on proteins and peptides. But basically, what I'm going to say will also, with some changes, apply to DNA and to other type of big, biomolecular objects like membranes or whatever. So I was hoping, I don't know how many of you are physicists or how many of you are chemists, how many of you are biologists, but I thought to make like a general introduction to proteins first. Okay, so why to study proteins? Well, proteins are the cell molecular machinery. Basically, they do everything that you have in yourself, and the functions at the end are going to be mediated by a protein. And in spite of this very general type of function that they can play, they are chemically really simple. They only expand, it's just combination of these 20 letters, of these 20 amino acids, that makes all the diversity of proteins that you can find. And the reason that this seems a little bit contradictory, that you have something that is chemically very simple, composed of 20 letters only, an alphabet composed of 20 letters, the fact that you can get this diversity is because you can combine them in many different ways, so that you can get very different types of structures depending on how many amino acids you put and how you arrange the sequence in a protein. So this diversity of structures lets you to different types of functions of these proteins. And of course, to understand how this happens, or what are this diversity of structures that you can find in proteins is the main goal of structural biology. Okay? The first landing mark in this was due to, or the first inspiring moment, that actually for quantum chemists is a really inspiring moment, was the work by Linus Pauling, who in 1951 discovered the first of these structural motifs that you can find in proteins by solving the X-ray structure related to alpha helix. He found this alpha helix motif. And he was able to do that by combining the realization of two very important aspects, very important structural aspects in proteins. One, the planarity of the peptide bond. The peptide bond is going to be this bond you form when you merge these two letters, these two amino acids. So he realized that that bond has to be planar. We have here a peptide bond, another peptide bond, another peptide bond, all of them planar. And the other things that he realized about the importance of what we call non-covalent interactions to stabilize these structures, in particular hydrogen bonds. So alpha helix is very stable because you are going to have all these hydrogen bonds stabilizing this spiral staircase. Okay, so the fact that you have a planar peptide bond also allows you to use something that we use a lot when we analyze our simulations, something that we call the Ramachandran space or the Ramachandran plots. Because basically, since you have a peptide bond that is planar, you only need two dihedral angles in order to understand how one of these peptide bonds, of these planes, locate with respect to each other. Okay? And we can represent this in what we call Ramachandran space. And what we see is that here we have one of the dihedral, on the x-axis the other dihedral. And if you plot what type of dihedral you find in proteins that they are folded, you see that you have like a disordered thing. You have like quite well-defined places in these Ramachandran spaces that are very populated. Of course, this corresponds to these what we call secondary structure elements, things that are quite stable in proteins, in protein structure. The one related to alpha helices, you find it here. This is the one that Paul discovered. He also discovered this or proposed this beta sheet structure, which they lie here. And actually, all the protein structures are a combination of this type of secondary structure elements. For example, here you have a protein that is combining two alpha helices with some beta sheets here and some loops that normally are disordered connecting these secondary structural elements. So this is what it makes this diversity of structures. The other inspiring moment in the structural biology, at least from the simulation point of view, came in 1977 when Martin Karplus, who was mentioned by Ronen and the person who I did my postdoc with, which actually was a former PhD student of Pauling. So scientifically, if I cannot allow me to say so, and the son of Karplus and my grandfather is Pauling, which is, but anyway. Oh, this is weird. So, well, let me come back to the talk. Martin Karplus, who ran the first protein dynamics, was almost 50 years ago. And with the computer they had at that time, of course, it was a very limited simulation, okay? And but it was really fundamental, as Karplus says, to change the view of proteins. Like until then, big proteins were seen as rigid structures. This is not strange because from the experimental point of view, until then, mainly you only have X-ray structures, which by definition are rigid crystals. And actually, we then have the tools in order to analyze the dynamics of a protein. And by this calculation, he demonstrated that they are not that rigid, the proteins, that actually they exhibit fluid-like motion, that these dynamics, you need to include it in order to really understand the function of a protein, okay? Well, how he did that? Well, he did that basically by simplifying a lot of the proteins, okay? So basically, we forget about quantum chemistry here or quantum mechanics, okay? We basically use, we take proteins to be classical particles that obey very simple classical equations of motion, and actually to calculate the energies and forces that you need to propagate this classical equation of motion, you take a very simple recipe, a recipe that is mainly formed by harmonic oscillators to account for the covalency of the bonds, the chemical structure, let's say, and very simple classical forces to account for the weak interaction. Like, for example, the hydrogen bond I was telling you at the beginning. But this very simple recipe is very useful because we can use it, we can propagate the equations of motion for longer time than if you will do quantum chemistry, of course. And it seems that for many things it works, even if it's a very simplistic view of what is an atom and what is a protein, of course. Well, even if you take this simple view, there are two things to take into account when you do this. The task is very difficult. This is something that you need to realize when you are simulating proteins. On one hand, proteins tend to have a small number of important states, not for the system we'll show later, but in general, okay? But to go from one state to another, it takes in the order of microseconds to milliseconds. This is an eternity, even for nowadays simulations. So transition between states are rare events, and you have to do something about it, okay? On the other hand, it is quite striking to know that everything is played in a very short free energy range. We are having, in these simple models, we are having thousands of tuple interactions, okay? But still, the difference in free energy between an unfolded state to a folded structure state of a protein is only 5 to 15 kilocalories. Just to know what that means, if you take the hydrogen bond, a single hydrogen bond between two water molecules, is already 4 or 5 kilocalories. So if you commit just a little error on that, you are really off if you are trying to simulate this type of things. Okay, so that's a difficult task. And the reason why this this scale is so short because you need to realize that you are always fighting entropy. So when you have an unbounded state, you have a sort of spaghetti-like state, okay? You have a lot of entropy. And in order to get to this point, even if you have a lot of two-body interactions that are stabilizing the system, you're going to have a very low entropy. So your entropy penalty, or if you want, your phase space volume here is very, very big, and here your phase space volume is very short. You have to find that in order to get an unstable globular state of a protein. So these two things are really difficult to take into account. Okay, so where are we? And even though it is something that a word of caution is that we need to take into account when we work with protein, but of course the field has advanced a lot. I think we are reaching nowadays, at least for short peptides, are reaching where simulation and experiments are finding each other in terms of time scales. And just to give you a sort of general idea of where are we now in terms of time scales, let's say that in the simulation, classical in the simulation, we have not considered explicitly the electrons, of course. This is the time step that we have to take, okay, in the order of 1 femtosecond, 2 femtoseconds, because we need to, this is detected by the fastest movement that you have, which is normally the carbon hydrogen vibrations or whatever with hybrid vibrations, okay? This was the time scale that calculus attained in 1977 in the order of picoseconds, okay? And nowadays, for short peptides and with some biased molecular eddy, we are already here in the order of microseconds. This means that we have billions of steps, or I don't know whether billions in this case is American or European billion, but it's 10 to the 9 steps that you need to analyze, 10 to the 9 structures that you need to analyze, which is another problem. Okay? Still, this is impressive. What we have done is million this in almost 50 years, very impressive. Still, you need to realize that this is still of marginal bio interest. Bio is played in longer time scales. Normally, here is when bio begins to be interesting, in the millisecond range. Okay, so we need to be a little bit humble. Still, we need to increase this a little bit, this frontier. Okay. Well, so how we have gone from here to here, and how we hope to go from here to there, how we hope to increase the time scale that we can afford in our simulations. Well, basically, there are four places where the people, the researchers, are doing a lot of advances, and that we need to combine normally when you do a simulation. Well, of course, hardware is important. In recent years, the fact that we have GPUs has really increased the time scale we can do. Of course, in the community, like in other, like everything in quantum chemistry, the people speak a lot about quantum computing. I don't know nothing about quantum computing. I don't know whether all the things that I have read now is something very general statements. So I don't know whether it will become practical. Okay. But what is really becoming practical is different things. Of course, I spoke a little bit about force fields and accuracy. There are all the time improvements on force fields, and include data from quantum calculations that are very accurate. Of course, it is a reality also machine learning force fields, which seems that they are changing the size of things that we can do and the time scales of the things that we can do. And also, it's very important to understand that probably if you want to say something of biological interest, you will have to use what we call enhanced sampling techniques. These are tricks we do to the dynamics, sometimes to the energy, or sometimes to the way we propagate the dynamics in order to accelerate the dynamics. Okay. For example, we used a lot in the group recently replica change, which is to run the dynamics, several replicas of your system at different temperatures, and then swap from one temperature to another. This allows you to go over barriers that otherwise it would be impossible to see in a microsecond time scale. Many times when I was telling you in my postdoc, I did a lot of QMMN. In QMMN, normally you do umbrella sampling, that is, you put an umbrella potential in order to force the system to go in the direction you want. And the other thing that you need to take into account when you run simulations, as I said before, is that you are going to have millions of structures to analyze. It's not as simple as to take, when you do quantum chemistry, of course, what is behind is very complicated, if you will, but you have one structure, two structures, three structures to analyze. Here you have millions of structures to analyze. So you need to really be careful to analyze trajectories. And here you have many techniques for which there is an option that you make, for which there is some problems and advantages and disadvantages of the option you take. But many times we reduce the dimensionality of the problem, we define a reaction coordinate, we do cluster analysis in order to define what you consider a structure, what you consider a state. That is not something when you have so many structures. Okay? Of course, to combine all this is not that easy, even if our view of what is a protein is very simplified. You need to combine all this, and it's not that easy. And in this sense, I want to acknowledge the IPC, because the IPC has supported two summer schools that we organized in 2016 and 2019. Actually, in 2016, had campus as well, joined it to this summer school, and many of the people, of the Grzelczak people that I overlapped with them in the campus group, they came, and we had this very interesting summer school in which not only we gave theoretical lessons about all these methods, but actually we also had in the afternoon hands-on practicals, so that the people could learn about this enhanced something techniques. I think actually Ronald also took part in this school. Thank you very much. We did the first one in 2016, the second in 2019. The third one we didn't do it because that was 2022. We all know the problem with COVID. But actually, this was the reason why we didn't organize it, because I became parent again, my second child, and I thought it was too much to organize in the school while I was in the kindergarten. So basically, we hope that in the future we organize more of this school, because the people was very happy with the result of these schools. Anyway, up to here, the general introduction that I wanted to do to protein simulations. I think I gave you the framework of where we move, the limitation of what we have, and what we need to combine. A way to analyze the trajectories, sometimes an enhanced molecular dynamics techniques, and always, the other message, be careful with your force field, because of course, a force field is very simple, very fast, but garbage in, garbage out. So you always need to be very critical with what you see in your simulation. And after saying that message, I want to move to the second part, or the third part of the message that I wanted to transmit today, and it gives you a little bit of a flavor of the things that we are dealing with more recently. I'm not going to go over the 4 different types of research lines that we have in biochemical, but I just want to focus in these systems, intrinsically disordered proteins. And the fact that I want to focus in the system is because, from the dynamical point of view, are the most interesting ones, or are the most difficult ones. Okay, so first of all, what are IDPs, intrinsically disordered proteins? Well, proteins are proteins that, in principle, they have energy landscapes that they don't appear to be very ordered. So before I was showing you something like that, a potential energy surface, where we have the unbounded state, unfolded state, so not unfolded state of a protein here, and then you have like something, a protein with a structure that is globular, that is very, very stable, etc., at this position. That corresponds to a potential energy surface of this shape. However, there are proteins or peptides or regions of proteins that they don't have this shape, but they have this another shape, a very shallow region with a lot of potential wells, if you will, a lot of conformation that is accessible at a given temperature. And this implies a challenge. On one hand, for theoretical methods, because here perhaps you could live with simulations that are shorter in time scale, but here you really need long-time simulations to cover all these shallow potential energies. But on the other hand, from the experimental point of view, also are very challenging, because normally you get crystals for this type of situation, and normally situations like this, they don't crystallize. Or it's very difficult to interpret whatever, NMR, for example, for this type of system. Okay, so it's very challenging from the experimental point of view and from the theoretical point of view to look at this type of system. And actually, in structural biology, they has been sort of put it aside. The people didn't want to look at this type of systems. Okay, but in the last year, we have learned that actually many of the proteins that are encoded in your genome are intrinsically disordered. It is calculated nowadays that 1/3 of the eukaryotic genomes is composed by intrinsically disordered proteins. So they are very difficult to analyze both experimentally and theoretically, but actually are very important. And actually they are behind of many things that cells that, and behind many diseases. The most known ones are the neurodegenerative diseases like Alzheimer's disease, but not only cancer diseases as well. They take part in many signaling processes, but doesn't really need to be related only to disease. It's something that the body is using all the time. Okay. Also, it's very important for liquid phase separation. There are many biophysics inside the cell that is mediated through this type of systems. In the group, we've been doing quite a lot of work related to different areas of IDPs. Some of them are related with neurodegenerative diseases. Some of them are related to protein recognition mediated by these IDPs. Others are mediated with developing non-natural peptides that they have some biological relevance. And even now we are actually starting a collaboration with Agune and with Juan Carlos, who is in the audience and a new postdoc of the group, because actually you can use this concept that we are developing here to understand how to immobilize enzymes, which is something not related to biology directly, but it has some technological applications. Okay, so I'm going to give you an example of each of this work that we're doing, except for this one. Okay, so that you can get an idea of how we analyze the system and what type of information that we get for these systems. So with respect to the interplay between IDPs, aggregation, Alzheimer's disease, and metals, basically this is what we try to analyze. Neurodegenerative diseases, perhaps the most well-known is Alzheimer's disease, is caused by the abnormal aggregation of IDPs. So you have these IDPs, and we don't know why, but they began to aggregate, and they form these toxic bodies around your neurons. There are two types of bodies. One is the amyloid beta plaque, and the other, which are outside the neuron, and the other are the neurofibrillary tangles, which is within, therefore within the neuron. Okay? And this is a very controversial issue. We don't know still what is triggering the formation of this. One of the hypotheses is that if you have metals in your brain, it seems that in people that has this problem, they have a concentration of metals higher that would be healthy. Okay, metal will bound to coordinate to these IDPs and change their structure and to take this potential their resurface, and in that way somehow trigger aggregation of these bodies and provoke these problems. Okay, so we've been working in different aspects of this problem, both with alpha beta amyloids and also with neurofibrillary tangles. I'm going to show you recent work we've done with neurofibrillary tangles, where basically in this case we took aluminum, which is, I don't have time to get into that, but it's one of our favorite metals in the group. Okay, and we analyzed how in aluminum will interact with short ADP segments, okay, present in proteins that are within the neuron. Okay, and normally this sort of aggregation, it is known that is mediated through phosphorylation. Phosphorylation means that one of your amino acids, for example, a serine, is going to be substituted by a phosphate group, and it is known there are several evidences that points that this phosphorylation, along with the interaction with a metal, is responsible for this aggregation. So we took this human missile neurofilament, that you can see the structure here. It has this long alpha ellipse. It's a very stable and very ordered part of the protein, but it has also all these sort of loops that are completely disordered, and actually a loop that is composed by this peptide here, the certain peptide here that is repeated several times. And this peptide contains two positions, two serines that are particularly prone to be phosphorylated. So we consider this type of systems, and we try to answer these three questions with theoretical methods. One, identify the proper coordination environment, whether, for example, aluminum or other metal will bind to one of these phosphopeptides, and which position and how within this coordination environment. Second, once this metal binds, how it affects the dynamics and the conformation of this peptide. That makes a chain, doesn't make a chain, depends on the coordination, depends on where it coordinates. And the third is whether we can have some information as identifying possible seeds for the posterior aggregation and the formation of toxic species. The first of these questions, the coordination environment, you can answer very, well, I will going to say very nicely, but you can answer by quantum mechanical methods, okay? Because normally that is a very local information. You don't need to treat the whole protein to know whether a coordination sphere is going to be very likely to accommodate a metal or not. Probably with directly coordinated residues to the metal, or perhaps with the second shell residues is enough. And that we can treat it very efficiently, very accurately, I would say, better, with quantum mechanical methods, where we take a cluster that considers the metal and the residues that are around the metal, and a continuum model to account for solvent, for example. And we can analyze like a lot of these sort of potential coordination sites and see which ones are more likely to get the metal. And I'm not going to describe you all the results, but basically what you see in this case is that, not surprisingly, whenever you accumulate 3 negatively charged residues around the metal, that is a very stable coordination environment. You put less, it's less stable. If you put more, the Coulombic repulsion between the metal is too high. So it seems that aluminum, and iron 3 as well, I would say, it is happy when you have like 3 negatively charged residues around. It's very simple. But it's a very simple recipe that we can check now many types of different coordinations for the 13 peptides. So the next thing we do is we go and we produce all possible combinations of monocoordinated peptide to aluminum, decoordination, tricoordination. In this case, for a certain peptide, we came up with 27 possible different coordination environments. We put the metal and we run classical ND simulation in the order of microseconds in order to check whether when you put the metal, the coordination or the conformation of your peptide change, whether it is affected. And to make a long story short, I just put this here because the simulations I'm going to show you, you need to realize that all of them are inside water. So part of the peptide you see, you have thousands of water molecules around that I'm going to put because it would be like difficult to see something. But don't forget that everything is happening. Okay. So to make a long story short, I'm showing you here 2 dynamics. Both of them have the same coordination, so locally they are indistinguishable. Okay. But as you can see, one of them you have like a sort of spaghetti-like movement, whereas in the other you have a really contorted structure. So in principle, yeah, aluminum can affect the coordinate, they can affect the conformation and the dynamics of the peptide, but depending, of course, in where you coordinate this aluminum. Okay. As I said before, you cannot get very easy conclusions if you just look at the dynamics like this. Okay, you need to do something to analyze the trajectories. In this case, we did some cluster analysis. I'm not going to get into details how we did these clusters, but basically we clusterified our dynamics with peptides with different degrees of contortion. And you can see basically this information of what I was telling you. So basically, each color represents one of these clusters. And if you have like the free neurofilament or the phosphorylated neurofilament but without the metal, you see a very wide distribution among the different clusters. And if you bound aluminum locally, you see that this distribution is not high perfect. However, if this aluminum begins to bind things that are like far away from each other or relatively far away from each other in sequence, you get this very highly contorted structure. Okay. Well, this long dynamics has allowed us as well to calculate something that I was telling you at the beginning. Calculated how much entropy you have to pay in order to get something that was intrinsically disordered, that was moving around. You put a metal and you get it very hot. So you can evaluate how much entropy penalty you have to pay in order to create that structure. And you can do that by information theory and by looking at the probability distribution of the dihedrals of the Xabier space. And I don't have time to analyze the theory, but basically from that probability distributions and from the joint probability distribution of the dihedral angles, you can get an idea of the entropy penalty that you have to pay in order to get this structure. So one of our conclusions was that we defined 3 necessary conditions for seeds to create seeds for aggregation. One, they have to have favorable conformational environment. That is something that we plot in this axis, and this means that your current calculations will give you a right coordination in the sphere. The higher you are in the sphere, the better. You have to pay a low conformation entropy penalty. The more you are to the right of this scheme, the better. And finally, your structures will have like an extended structure so that it can aggregate to other fibers. And most of these structures do not feel that, but we are able to identify some of the structures they do fit these three conditions. Okay. Next, what we are trying to do is calculating ligand, for example, and we are trying to accumulate more fibers. But for that, these methods are not good enough, are not efficient enough. And one of our students, David Silva, is now doing an stay in the group of Professor Cecilia Clemente to learn how to use coarse-grained machine learning force fields in order to be able to expand the limits of what we can do in this area. We also have been looking at other systems for which we have more experimental information. And in that case, we have alpha-beta amyloids bound to think that you do have, for example, NMR. You have a lot of NMR data, a lot of structures, you have N-oid spectra, things that you can compare with in certain sense. And just to make a long story short, in this case, we learn how to incorporate that experimental data in our simulation. You can do the same step I was telling you before, analyze the details of the coordination with web methods, transfer those details into the force field, creating bonding models to think for this different type of thing coordination, run long-time simulation, but apart from that, what we can do now, since we have experimental information, is reweight your dynamics in order to incorporate that experimental information. And you can do that by what is called Bayesian maximum entropy minimization, which basically you are, after your dynamics, you are reweighting your dynamics according to this formula, which each square will be the difference you expect experimental data, and this entropic term will be like a penalty that you put in order to not depart too much for your initial weight. Okay? And to make a long story short, what we get combining these methodologies are ensembles of this thing that are similar to the assemblies that we are looking with, I showed you before, with aluminum and neurofibrillated rectangles, in the sense that you get ensembles that they are very, very corresponding to what you find in IDPs, where basically what you need to combine is contorted structures with extended structures that are possible to aggregate. And this information is something that you cannot retrieve from PDB, because sometimes PDB, they give you very sort of, in the way they solve the PDB, when you do NMR, they give you normally only the contorted structures. So this is a way also to improve your dynamics. You can put in experimental information into it, okay? So what we have learned in this area about how metals interact with short peptides, we have been using to analyze, to design antimicrobial peptides. In this case, I'm going to be very short. Basically, the idea is there is interest in, as I told you before, proteins are words combined by 2 letters, 20 letters, right? We have 20 natural amino acids that form proteins. Well, we will include also in amino acids things that are not encoded in this alphabet. For example, we have myosin, which is a natural amino acid, but it doesn't normally, no, it doesn't normally, no, never. It is not encoded in the genes, So it doesn't form part of our protein space. And what we can do is to generate IDPs composed of these mimosins, and in principle, these non-protein amino acids, they have seen that they have significant biological interest because they show antimicrobial activity, basically because of chelation ability of metals. Okay, so an experimental group contacted with us, telling us, just asking us very simple question. For example, what is the sequence that I can prepare so that I put 3 mimosins there and I collect the metal? Okay. So we're using the same methodologies, QM, to analyze how these things get coordinated. Running the QM, we can develop new parameters in order to account for mimosin within the force field that already exists, and also to account for the meta-mimosin interaction, running on time simulation and getting our idea about the configurational entropy. We could propose which sequence we thought would be ideal for metal chelation. Okay, and we propose this sequence, a sequence that should be composed by 8 peptides, a proline in the middle, and actually this was synthesized and by ultraviolet visible spectroscopy, they detected that actually it was very good collecting method and actually has antibiotic effects because of this chelation. So this is an example of how all these theoretical things we can use it to give an answer that that experimentalist proposed. And the final project I'm going to show you is another project in which an experimental group has proposed us something. Okay, and it's the one related to MCL1 PUMA recognition. So MCL1 PUMA recognition is an important target for cancer disease. What happens here is that, of course, biology is very complicated. You have many cascades of reactions possible, but at what point when your cell detects that it has some damage in DNA, it began to express PUMA. PUMA is this protein that we see here in blue. Okay, so when PUMA is being expressed, it began a cascade of reactions that in the end give a signal to our immune system that cell, something wrong with it, and you need to get into apoptosis. Of course, cancer cells are very smart, and one strategy they devised to avoid this apoptosis is to express MCL1, which is another protein, which is here in white, that has higher affinity for PUMA than BCNL. Okay, so you get this interaction here, and this is a way that cancer cells have to avoid this apoptosis. Okay, so you have here a little bit more in detail how this recognition between PUMA and MCL1 works. PUMA, or the domain that we consider PUMA here, is an intrinsically disordered protein. The type of system we're interested in. So it doesn't have, it's like a spaghetti. But once it finds MCL1, it produces this, what we call a hot loop, this little alpha helix, and this little alpha helix binds to the proper place. And because of this binding, now the rest of the helix is produced, and then it's going to be, the binding is going to be very strong, and that is the way that you're going to avoid to go over this path. So a group of experimentalists contacted with us, the group from the University of Copenhagen, Professor Rogers and Fabian and him, but basically the idea is to generate peptides, okay, where basically they cyclate the peptide. They make a peptide with a cycle here, with a ring. And the idea is that this ring is going to mimic this hot loop. You don't have to pay the entropy penalty associated to the formation of this loop, and it has a higher affinity for MCL1 than the natural PUMA, and you can avoid, basically what we are going to do is saturate MCL1 with this cyclic peptides. The important aspects to be considered here is that you need to create a ring, and this ring has to be, or has to promote an alpha helix in this position. Okay, so they run this experiment, they produce these peptides, they run many different experiments, they play with two peptides. I'm going to show you only one because it's the same philosophy with the other one. We have the same results or similar results with the other one. And basically, they were successful in the sense that they compare with circular decreasing data. You can estimate the alpha helicity of your peptide, and you can see that the Eliot, we call it like that, to this bring here, the Eliot peptides, they show a higher helical content than the linear one. So it seems that it's acceptable, their strategy. And the other interesting thing is that this alpha helicity that is provoked by this ring, it has a huge chiral effect. So if you introduce in the first amino acid L amino acid, which is the normal enantiomer that you have in proteins, you don't have any effect. Basically, you have the same helicity that you have in the linear compound. But if you introduce at the first amino acid a D amino acid, is when you have this helix form. And that's because they didn't understand why this is happening. Okay. So in this case, the methodology we use several force fields. As I told you before, you have to be really careful with the force field you use. Okay. In this case, we use this force field developed in 2019 by Robustelli, Shaw, and Viana, where basically it has been parameterized taking into account not only folded proteins, which is the normal situation for the normal force fields, but an amazing amount of data from folded proteins to disordered proteins. So it gives you a very balanced description of both situations, folded state and unfolded state. And we couple this with one of these enhanced molecular dynamic simulations I was telling you, replica chain molecular dynamics, where basically we run the simulation at different temperatures, and once in a while we swap between the different temperatures according to a Monte Carlo criteria, so that our replica that is at the lower temperature at infinite time, it will recover the canonical distribution. And these are the results. Basically, our empty simulations accurately capture the tendencies to form alpha helices. You have here the fraction of alpha helices we see in our simulation for the Eliot motif, the cycling motif, and for the linear ones. So in the linear ones, almost you don't have any helix, but in the Eliot ones, you do have helix. Basically, focus on the ring will be here, but that propagates partially to the residues that are nearby. This you can check also with, we can compare this data with secondary chemical shift from the NMR experiment, and we got a reasonable agreement, except for one outlier, okay? And so that's in certain sense support our in the simulation. And now we can analyze our simulation, and what we see in the Eliot motif, if we analyze the hydrogen bonds that you can form within the ring, between the third peptide, the fourth peptide, which is this peptide within the ring, and the peptides that are outside the ring, we see again that we have this E+4 hydrogen bond system that you so stabilizing in an alpha helix. The same was that Pauling was looking when he discovered the alpha helices. We also recovered the chiral effect. So this is what you observe for, and we ran a simulation with D amino acid at the first position and with L amino acid at the second position. And you can see, as the experiments will show, that basically when you have the D cycle is when you have the signal we were seeing before, is when you have this hot loop created. But if you put the amino acid in the L position, the signal is destroyed. Basically, you don't have helicity. We can visualize this in a more visual way, let's say, by looking at the ring conformation. In order to see that properly, or more easily, we just get this reduced model of what is the ring, just beating the alpha carbon, and you see that in the D version, you get a very contorted ring, which is coherent with an alpha helix, whereas in the L version, the ring is very planar, and you destroy the alpha helix. And to understand this, we went to the Ramachandran space. This Ramachandran space I was telling you that is important to understand the conformation of your peptide. And when you see that basically the L-phenyl aniline and the D-phenyl first residue, they are sampling very different regions of the Ramachandran protein. One is here, the L1, and the D is this one, which is normal, or is what we expect from L to the enantiomers. This is what I'm putting here is what you expect for an enantiomer. Normally, you will be something in this region of the Ramachandra space. In the enantiomers, you will be in this area of the Ramachandra space. However, they both is not the mirror image of each other, because in one case you are sampling close to the alpha region, in the other we are sampling in a region that is called the polyproline 2 region. Okay? And to understand this, basically what's going on, we create this sort of photo models, in which one, in the one here, we are creating an alpha helix with the first residue, the backbone of the first residue in this position. And you get this torsion of this peptide towards downwards. And this position here is fully compatible to create the cycle, because this is the bond that you need to make in order to create the cycle. So the ring is fully compatible with an alpha helix. However, in the case of the L amino acid, if you create a perfect alpha helix, this sulfur that has to cyclate into the carbon to create the ring is too far away. So the system with the L enantiomer, the only possibility you have in order to create the ring is to get it more planar. You need to destroy your spiral staircase in order to be able to plug to make this. Okay, and that is the origin of the reality that we observe in this system. We were lucky that also have no structures, NOE structures that we could look at. They gave us 20 structures that came from NMR that we could take. We can optimize with QM, because here we trust more QM. We optimize with QM, and we see for the most stable structures the same structural motif that we are looking in our dynamics. Basically, the first residue in polyproline region with very nice hydrogen bond between the third and the fourth peptide, and for the rest is alpha helix, stabilized by the same hydrogen bond prediction from empty simulation. Okay. So next, what we are trying to analyze, we're trying to understand, is how these peptides bind more effectively to MCL1. And to do that, we are creating structures departing from alpha hot, because they are not experimental structures, where we put the PUMA, the original system, we put our cyclic peptide here, and we are in collaboration with Julia Contreras, who was visiting the IPC last month. We are looking at all the non-covalent interactions that are responsible for this stabilization and comparing them in order to provide to the experimentalists with possible mutations that increment the binding. Okay. So I guess I give you an idea of this work we did. Basically, to conclude, I would say that after almost 50 years in the first protein simulation, we are living in a promising era. A lot of challenges, but we are reaching an era where we can compare with experiments, actually, because we are in the same, if you do enhanced molecular dynamics, you are approaching the same time scales, okay? And if your system is small, let's be honest with that. I think I showed you that simulations can shed light on behavior of intrinsically disordered proteins or peptides, which are very important, are very difficult to analyze both from the experimental point of view and from the theoretical point of view, but they have a big biological relevance in many different fields. Okay. Enhanced NT simulations are needed, and new force fields, of course, always are needed, are welcome, but you always should be careful. Garbage in, garbage out. And force field can give you, I didn't show you the results before, but we tried other force fields, and sometimes you get a perfect alpha helix when it should be an intrinsically disordered peptide. So you have to be very careful and incorporate the data from 2M calculations as much as possible in your force field. Okay, this is especially relevant when you have metals. Okay. And when I hope I convince you that we can provide for some rational design of peptides that they have biological relevance using theoretical methods. And just to finish, I would like to acknowledge basically today the work I presented was the most recent work is due to very talented students, two very talented students, Julian Ruiz, who has also the nicest picture here, and David Silva, who is the one that is now in Berlin, working with post-grain machine learning force fields. In the past, also Rafa, who was a former postdoc student of the group, also contributed a lot to neurophilia entangled formation studies. Gabriele, Johnny was a postdoc in the group, did a lot of work, was a great scientist. I am very sorry that he quit science nowadays because I think he was a very talented science and he contributed a lot to the things that I mentioned today. And Elena, of course, who is in pharmacy, in the pharmacy school, is a professor that he did a postdoc with one of the main persons in this area, Marinello, and she also has done a lot of work not in the part that they showed today, but mostly in the simulation of other type of systems, enhanced molecular dynamics techniques. I will also mention that we have done collaborations with agents of the vaccines and technology network. For example, last year we presented two theses in collaboration with Tanogune in the field of DNA, which I didn't mention anything today, with Piomagune and with Piomagune. And that what we are doing is we are using a lot of techniques that, of course, something we need to learn from other people. And we are collaborating with a lot of experimentalists. And the work I was showing you today is part of a collaboration with different institutions. And I thank all of them as well for their creativity. So anyway, I'm thankful for you for that.
Well, thanks a lot for very nice and exciting presentation, and also reminding us that what we are doing is not always useless.
It was the fancy way.
Time for questions. Yeah.
Hi, Xabier. Very nice talk, very clear. So I have two questions. So in the beginning, you introduced this Ramachandran space, where we have these two concentrated region, alpha and helix and beta sheets. I also know the third isolated region on the right.
Yeah.
What is that? Is there any?
This is an alpha helix that normally alpha helix made from L amino acids, they tend to the right. An alpha helix made from D amino acids, they tend to the left. It's kind of right now. But I mean, this is not like there are special systems or special sequences that sometimes with N amino acids you can turn also to the left. But it's not very highly populated. It's like sort of the special sequences are authentic. You have to combine many proteins or many, I don't know, and then you get like for special systems you get in those regions. Okay.
It's still alpha helix, but opposite.
But to the other side. But it's not very highly populated. I don't know whether you remember this, but the blue color was really light. So it's really sort of not the common conformation that you see with elements. Yes, with the amino acids is the mirror of it.
Okay. So second question, I'm not expert in this simulation, but I was just curious that in your simulation, did you put the protein sort of like a simulation cell?
Yeah.
And you said the temperature, you said the pressure. So what pressure?
Normally, well, you run like different equation steps depending on what you are looking at. But normally we run a elevation step, for example, in MPT ensemble, which means that we set the pressure normally to 1 atmosphere. The temperature depends. In the last case that I was showing you, these peptides in the experiments were done, I think, at 283 Kelvin. We choose that temperature. But normally we run at 298 Kelvin. That is the sort of the standard temperature. But we combine different steps, like equilibration step, like MBT ensemble, then MPT, then have MPT, and then you run the simulation. Depends on what we are looking at. Sometimes we use the canonical ensemble, MPT, and sometimes we use MPT.
Thank you.
Yeah.
Amyloids. So metals, no metals, it's still not clear, I guess. Of course, as an inorganic chemist, we would love metals would be fundamentals of the most stuff to study. But the way they assemble, for example, metal or non-metal, seems random, right?
Well, I mean,
Yeah, you show in your simulation, if I put in the spaghetti there, spaghetti, if I put it, then but biomolecules, there's no reason why you should go there. Or is anyone studying any protein-assisted mechanism for this type of things, as you showed for Puma and so on. So a protein coming and helping.
I know, for example, the catalytic antibody that it tried to destroy the fiber. But once it is formed, and this was one of the most promising therapeutics of the field that has been quite nowadays. It didn't give the results that they wanted. I mean,
But in the mechanism of formation,
in the negative, I haven't seen it. And I have to tell you, so about the metals, I didn't have time to the mechanism I was showing was like you have the metal, your spaghetti is moving around, you get the metal, and somehow this gets a more extended structure, and then it's easier to get. This is the metanucleation hypothesis. I think that probably the other hypothesis with metal is like first you get the fiber formed without the metal, and then the metal binds and stabilize a fiber. I think it's more likely. And we see that because I show you about all these different points. Only one will act as a seed for recombination. Mostly the other ones not. And still you have to pay a big entropy penalty in order to accommodate a metal there. But if you have your fiber already formed, your fiber is already quite stable. And to introduce a metal, you don't pay the conformational penalty, but you provide the same coordination environment. So it's like, and I think that is more like, it is true that in brains with people with heart disease, they have a concentration of metal. For example, zinc is 200 times higher the concentration as a normal brain. But there is all this controversy whether this is a consequence or a cause. And I don't think that really people still know, which give us a lot of places of explanations and
not that I know much about this, but there is this AlphaFold consortium to predict the structures of 3D proteins.
Yes.
First of all, maybe you say one or two words how that works. That seems to be tremendously successful.
Yeah.
Secondly, I guess that works in the case of what we call disordered proteins. But what happens if you have this intrinsically disordered proteins?
That's a very good. That's a very good question. Actually, the last structures I show you, if I can put them, yeah, this one were created with AlphaFold. Okay, this one here. And first of all, AlphaFold is really a revolution in our field. To take into account, before when I was in the calculus book, the first step you need in order to begin your simulation is whether you go to the PDB, the protein data bank, where you have all these structures, and if you wouldn't have a protein there, you could not start your simulation. So you were always expecting a structure, you need to have an experimental structure to start your theoretical calculation. This, with AlphaFold, this is a revolution. Now we have as many structures as we want. Now the question is, are they accurate? For globular or for ordered states, this is very, very accurate. But of course, this is like an extrapolation between whatever you have in your data set. So the disordered proteins, how they appear in AlphaFold. Well, AlphaFold, when you put your sequence in AlphaFold, it gives you an structure, and it gives you also the residue, a sort of reliability index. And we think when we put intrinsically disordered proteins or regions of proteins that we know that disordered, is that it's going to be something that can do like that. Yeah. But it's good to tell you that that region is not very related. It's already giving you information. Okay. So it is also useful in this sense for but it's really has really amazed us now that well, the protein space is actually very simple. For me, was okay, AlphaFold was successful, but in the end, proteins have all these secondary elements that you put together and they cannot put calculus. But what is impressive in AlphaFold 3 is that they can go through the chemical space. Now they can actually provide you with the structures that bound to different ligands. And the chemical space is much more complicated. So I don't know how well the efficiency they have, but they claim that this is going to revolutionize as well the field of voting or of other field that is thought it would be like outside the machine learning to do. So yeah, No, it's really a revolution.
Sorry, these alpha helices that you intentionally use there, are they D or L or racemic?
Now, the beauty of this, or at least for me, the beauty of this is that is this first residue the one that needs to be D. The rest of D is L. Of course, if you will create a peptide with all D amino acids, you will see a change with respect to all L amino acids. It's just a mirror image. It's when you combine D with L that you begin to see these effects. Okay? Yeah, so it's the first one that is D. Okay? Yeah, I don't know whether I answered your question, Jesus, or
Well, in a way, but I was more looking at the helix that this produces in the other protein whose name I don't remember at the time.
okay.
So you
You mean the one with alpha 4 that I'm pursuing?
yeah,
Okay.
probably. No, this is too big for me to grasp. This include there.
This is all L. This is the natural protein. Natural proteins only have L amino acids. So to introduce the amino acids that we can do to see whether we get advantage, but nature doesn't know how to put the amino acids inside the protein. So this is pure L amino acid. Okay.
And where's the D amino acid there in this picture?
Here it is not because it's a natural protein. Here the amino acid will be, I think, I don't know whether it is this one or this one. No, it's this one, the first one. This is the cyclic peptide.
Yeah, but there's still this peptide in blue is one of them, DOL, right? But you never get a combination of the two.
What? Sorry, I don't understand.
This blue one,
This one is the peptide. Yeah.
yeah, it is.
This is the DNA. The first residue, which is the amino acid.
But the
Elasticity, is it?
No, the
that-
acid is the normal one because the rest of the, I mean, the point is the following. Let's say this, and we can see this in sort of Gedanke experiment that I did with this alpha. So this first residue is here, okay? This is the D amino acid, okay? And the rest is L, so the L is on the right,
Left-handed.
right-handed. But the point is that the first one is D. So it doesn't sample in that area. It's sampling the proline region. So it's looking down, which normally, if you have an alpha helix, everything should look up. And it's actually that change which is really important to stabilize the rest of the alpha helix. It's very counterintuitive. I mean, it took us some time to realize about that. But that is the effect.
I understand now. It's the helicity and the chirality is as well.
Yeah, it's like you need to have the normal alpha helicity compatible with cyclization. And that is giving you, by the first amino acid, to put it in the D enantiomer rather than the L enantiomer.
Yeah, okay. Tell us one more thing is that you keep telling about or talking about this entropy penalty. All right, but you are doing molecular dynamic simulations. Entropy is not accessible from molecular dynamic simulations, isn't it?
Well, depending on which type of entropy. I mean, there's a very old paper of Carplus. I mean, in the end, you have to compare this more like unfolded folded state, right? I mean, trying to compare the entropy between something that is disordered and something that is ordered. And of course, entropy, you have many different aspects. You have the bonding terms, the angular terms, but all these terms, they tend to annulate when you are comparing the one that is ordered to the one that is ordered. But this the main signal of your entropy is how the way you see this, how your phase volume gets reduced when you go from a disordered state to an ordered state. And that phase volume we can find it if we look at the paramount, because it's what is giving you the relative orientation of one amino acid with respect to the other. That's why we use the probability distribution of this dihedral that comes from the paramount. So we are, to make a long story short, we are just looking at the contribution of different configurations to them. What we call the configurational entropy. But within is the main sign. Within is the main sign.
Yeah, but this is not the thermodynamic entropy. This
Yeah.
is the information that you receive.
Yeah, exactly. Yeah, yes and no. It's true. It comes from information theory. We are not doing like in the quantum practice of instigator and blah 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 and partition function, blah blah. We are not doing that. But it corresponds quite well to based on experiments what you expect for this like estimation from experiments of how much conformational entropy you pay per residue when you photon. This is between 1 to 2 kilocalories per mole. And this is more or less the numbers that we are looking at with methodology. So indirectly, you are probably right, but indirectly we think, and actually I don't have time to show you that. But I was dying for the opportunity to show it. This is what the observers experimentally. They have measured the difference in affinity between the linear and the Elliott cliff, and this is around 2 to 3 kilocalories per mole, which is very coincident with the configurational entropy estimation that we are giving. I mean, perhaps we are jumping too fast to the conclusion, but we think that actually this increase in binding comes actually from this decrease in configurational entropy that you get when you have a pre-organized cyclic peptide that you already have this contact group created. But this is to jump to, I was just finding discussion with this one, but we didn't put that in there because we are still thinking about.
Any more questions? As far as I can see, there are no questions from the people who are with us but are not present here. Yeah, there is a.
And thanks for that talk. It was very interesting. Regarding this point of whether one can calculate the entropy or not. Now, if you have exhaustive sampling and you're able to calculate the free energy, in principle, I mean, getting the potential energy is triggered and you should be able to access the entropy. Do you do that? Do you perhaps?
In principle, they can, yeah, I mean, you're right, people will be living here, right? At the end of the day. I have two answers to that. The answer